Welcome back to microbiology. Today we're going to be covering chapter 6 over microbial nutrition and growth. So to give you a brief overview of where this chapter goes, we're first going to go over what is microbial nutrition, what sort of factors are required for microscopic organisms to live and thrive, the environmental factors that can influence microbial growth, and how we can study these distinct parameters that certain species have. Some prefer certain temperatures, pHs, certain nutrients, and how we can study and tease that out. But for today, we're first going to cover this first bullet point of microbial nutrition as a whole. All life requires a constant influx of certain substances, whether it's a fish that we're studying, and houseplant, ourselves, whatever it is, there is a distinct requirement of certain nutrients that are required for survival, and these come from their habitat. All living things require a source for carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, sulfur, calcium, iron, sodium, and so on. These are essential nutrients. These are any substance that has to be provided to the organism. As in, if we don't have carbon, we're not going to live another day. We don't have oxygen even for a couple minutes, it can be life-threatening. Therefore, these are essential requirements for life. Now, it is important to note that they're not all required in vast quantities. Some are macronutrients and some are micronutrients. So macronutrients are any substances that are required in large quantities. So you can things like, yes, I need a lot of oxygen every day, all the time, or I need lots of hydrogen or carbon. Think nutrients that maybe you've seen in cell structure or metabolism, like what sort of molecules make up proteins or DNA or lipids, things like that. Those would be macronutrients. Micronutrients, on the other hand, while still necessary for survival, are required in much smaller quantities. So this can often include small ions that are needed as cofactors for enzymes to function, things like magnesium, maybe something like that. Molecules that help maintain or get proteins to fold properly, like chlorine or iron, things like that, but they aren't going to be needed in vast quantities. So to give you some ideas of the overall percentages of what you might find in a cell, we'll find the most abundant organic compound in the cell is going to be proteins. And that's because we find them pretty much every part of the cell. In the enzymes that help break down broken organelles, we find them wedged into the cell membrane. We find them in the replication machinery, in transcription, in translation, in the cytoskeleton. We find them almost everywhere. The next category that is also found in very large quantities is nucleic acids. So I find this one kind of interesting. It's different than what we typically think of from our biological understanding, but you have about half as much RNA as protein. So you have about 20 is roughly the amount overall here, which is much larger than the DNA. So DNA might be more stable and more stagnant, but we still have a lot of RNA being produced. You'll also find carbohydrates and lipids. So thinking like phospholipid bilayer, sugars, things like that needed for metabolism, and overall like miscellaneous molecules. This could be things of like those interesting enzymes and things like that. You'll also find a significant amount of water that's not included in this dry weight. If we further break it down, you can go through element by element. We are carbon life forms. If you're into sci-fi and things like that, we are carbon life forms. Carbon is the primary element that we're composed of. And then we go down the list. Oxygen, nitrogen, hydrogen are all required in very vast quantities. And then you get further down, and like calcium would be more of a like micronutrient, where it's needed much less than carbon, but it's still required. If you further analyze the cytoplasm of E. coli, for example, for its cell composition, you'll find that about 70% of the cell is comprised of water. So again, another thing when we think about what is necessary for life, yeah, we need water. And if you're stuck in a desert or your poor plant gets left alone for a couple weeks, you can tell. So all cells need water in some capacity. Proteins, as we saw on the last chart, are very prevalent as well. And your book gives this number, 96% of the dry weight of the cell. So if we dehydrated the cell, got rid of that 70% water, what was left behind is composed of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So those are going to be some of those macronutrients. Most of the quote chemicals in the cell are actually going to be compounds. So they're going to be things like water, which is, we know, H2O. So it's hydrogen and oxygen, not just a pure element. Even simple cells, so like our example of E. coli, have roughly 5,000 different compounds, which is just mind-blowing when you think of how many different compounds and then how many of those are duplicated. And maybe you only have 5 or you have 100 of a certain compound, and the numbers game can get really out of hand really fast. It's pretty miraculous. So moving on to, we have our basic standard of what a microbe is, what nutrients is required. But where do they actually get those nutrients? Specifically, where do they get the carbon necessary for survival? And there are 2 main paths that microbes can take. They can either obtain carbon from an organic form, so it could be like eating other microbes and taking them in and kind of using their organic substances and as their own. Or they can obtain them through conversion of CO2. So they can take carbon dioxide from the air and use that as a carbon sink. Heterotrophs get them from organic matter. Autotrophs obtain them from CO2. Another category that we can use is where do they get their energy from? And again, they have 2 paths they can take. They can either create their own energy from sunshine. Okay, so that would be like my picture up here. If you have like a succulent, they would be getting their energy from the sun. That would be a phototroph. Or you can have a chemotroph, which gets their energy from chemical compounds. So that would be like us, where we eat our delicious salad and all of those sugars and fats all get broken down, and we can go through a bunch of metabolic pathways to create energy. Those would be chemotrophs. So we can kind of mix and match those. There's 4 main categories that come out from it. We can have photoautotrophs, so their energy comes from sunlight, but their carbon source comes from carbon dioxide. This would be things like algae and plants. You can have chemoautotrophs, so these ones get their energy not from the sun, but from organic or inorganic compounds that they're going to be eating and kind of piecemealing together. And then their carbon is still from carbon dioxide in the air. So those are our first 2. The other 2 we can have are photoheterotrophs. So they're still getting energy from the sun, but their carbon is from an organic source. And we have chemoheterotrophs, would be ones that are getting their metabolism and their energy from maybe dead organisms or from a host like a parasite, things like that. And then their carbon source is also organic. So you can kind of mix and match. They could be the same, they could be different. So again, photoautotrophs are going to be photosynthetic organisms, organisms that can do photosynthesis like these cute little baby plants over here, capturing energy from the sun, transforming it into chemical usable energy for cell metabolism. They are the basis for the food web. So if you think back to Bio 2, you're going to have your algae or your plants, and then they're going to get eaten by small organisms that are then going to get eaten by a little bit bigger organisms and bigger and bigger and bigger until you get to the top of the food chain. Chemoautotrophs can either be chemoorganic autotrophs using organic compounds for energy and inorganic for a carbon source, or they can be lithoautotrophs requiring no sunlight, no organic energy, only inorganic material. And I have rocks down here at the bottom because there's some really interesting lithoautotrophs that are microorganisms that actually eat stone or rock and can convert that into energy and a carbon source, which is pretty miraculous, pretty cool. Most heterotrophs are microorganisms are chemoheterotrophs, meaning that they're going to get their carbon and energy from the same source, from organic compounds. These compounds are going to be processed through cellular respiration, fermentation, metabolism, things like glycolysis, things like that, to get ATP out, adenosine triphosphate, the energy currency of the cell. Chemoheterotrophs come in two varieties. They can be saprobes that are free-living microorganisms that are going to be eating dead organisms. So that could be things like maybe mushrooms growing on decaying leaves in the field or in the forest, things like that. It could also be parasites. So these would be organisms that get their nutrients from a living host. So this would be things like if you had a yeast infection or a urinary tract infection or a gut imbalance, microorganisms that are getting all their nutrients from their host. Parasites can live on the body, so that would be ectoparasites on the outside, so like skin, things like that. Inside the body, so that would be like in your gastrointestinal tract, something along those lines. And there can also be intracellular parasites or microorganisms that live within the cell, because again, the microorganisms that are like bacteria are going to be much smaller than the eukaryotic body cells within us. Obligate parasites are often very difficult to study because we can't grow them very well. Obligate parasites require a host to be grown, and I don't see anybody volunteering, for, hey, pick me, I'll take one for the team, you can grow some leprosy on me. That's not probably going to fly anywhere, which also makes it really difficult if we want to study them in their normal environment. Okay, so that's one big hurdle when we talk about microbiology and human health is how can we study these really difficult obligate parasites, because they are a public health concern, but we don't have a great way to mimic their hosts. Okay, so some example pictures here, cyanobacteria, these lovely little green bead chains up here would be a photosynthetic autotroph. Okay, so they're using sunlight and getting energy there. And we have E. coli down here at the bottom, these nice little gram-negative rods, and they're going to be chemoheterotrophs, getting no sunlight for them. They're actually going to be having to eat something to get their nutrients. Okay, so again, just a reminder here, essential nutrients, they're trying to get carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur There's just a variety of ways to go about it. Are we getting it from sunlight? Are we getting it from other organisms, from a host, from decaying matter? How are we going to get these? And these are essential for, if you go look at the chart here, for a variety of cellular everyday activities, from making proteins or lipids, nucleic acids. Oh, we got to have carbon for that. Hydrogen, it's going to be found in a lot of the gases produced by microbes. It helps maintain pH. Oxygen, we're going to be finding that one in a lot of the key biomolecules, such as proteins and nucleic acids. Nitrogen, found in all DNA, RNA, ATP, those nitrogenous bases maybe that you've heard. Phosphates are the phospholipid bilayer, the phosphate backbone of DNA. We need a phosphate to make that happen. So you can see how they could be very essential for these key steps within the cell. Okay, so just another way to look at it: potassium, necessary for protein synthesis. Sodium is an ion we use for cell transport because it's charged. Calcium stabilizes the cell wall. Magnesium is a component of chlorophyll for light harvesting. It stabilizes ribosomes. Iron is found in a lot of the cytochromes that were used in the electron transport chain. So when we're passing the protons down the electron transport chain to get a whole bunch of energy out at the end, we need cytochrome proteins, and they need iron to function. Or zinc. Zinc helps regulate genes in eukaryotes. So how overexpressed or underexpressed is a gene is often determined indirectly through zinc. So all of these are very important nutrients within the cell. Overall, survival of a cell, whether it's eukaryotic, prokaryotic, single-celled, multi-celled, requires that the organism is taking in the proper nutrients and putting out its waste products. The cytoplasmic membrane, the outside phospholipid bilayer on the cell, directs this movement of taking in nutrients and putting out waste. The cell wall is not selective like the cell membrane. The cell membrane is the thing that determines what gets through and what does not. Diffusion is the movement of molecules from an area of high concentration to low concentration. So normally, molecules are trying to get themselves evenly distributed. So we're going to want, we have a whole bunch of red molecules on one side and none on the other. They're going to want to even it out so they're 50-50. So often we see molecules moving across the cytoplasmic membrane simply by diffusion. No energy required. Not only do we see biomolecules moving, but we also see movement of water. And the movement of water, or the diffusion of water, is known as osmosis. You can have 3 different types of solutions when we talk about movement of water. You can have hypertonic, hypotonic, and isotonic. Hypertonic is when the environment has a higher solute concentration than a cytoplasm. So we have just a couple of keywords. We have the solute would be the molecule that's diffusing or dissolved in the solution. Okay, so if you have more salt in one area, then it's going to want to move. Hypotonic is when the environment has a lower solute concentration than that of the cell. And isotonic is when they are equal. So we can look at it a little bit more here. Isotonic is when the amount on the inside and the amount on the outside is equal, and therefore it's not stagnant. Some molecules are moving in, some are moving out, but they're pretty equivalent basically. So the rates of diffusion are equal in both directions. With the hypotonic solution, we have more of the little solutes inside the cell, and therefore water is going to want to come in and kind of dissolve it a little bit more, dilute it. This often leads to the cell swelling and even bursting. For whatever reason, I know it's a silly way to think about it, but I remember hypotonic because I think the first part is like a hippo, and hippos are mammoth and they could pop at any moment. I don't know. Okay, so it's a silly way to think of it, but it works for me. Hypertonic is the flip of that, where the cell shrivels up and shrinks because all the water inside goes out to further dilute the salt solution outside the cell. This leads to plasmolysis, so you'll kind of get a shrivelly cell, even though sometimes the cell wall stays put. So the shape may look the same, but the inside will be kind of crinkly. Simple diffusion is limited to small non-polar molecules. So the phospholipid bilayer that makes up the cell membrane is selectively permeable. It won't let some things through, but it will let others through. So molecules that can move freely are often like gases, like oxygen, and very small non-polar molecules. The cell membrane is going to be impermeable to very large ions that have lots of charges on them, and polar molecules, big molecules. We don't want our DNA just floating out, okay? So there is a size and charge requirement to get across the cell membrane. Now, cells do have mechanisms to help get around this problem. They can do active transport, or they can move molecules across the cell membrane by inputting energy, or ATP. Okay, so these are the three main ways that cells can transport things across the cell membrane. You have simple diffusion, so there's no energy requirement. It's just that it's a small nonpolar molecule and it can fit through the cell membrane and go back and forth. It's just going from a high concentration to low concentration. Facilitated diffusion is when a molecule will bind to a receptor and it will help carry it across the cell membrane. Okay, no energy required again. The final one here is active transport. Okay, so you still need a protein to help move it across the cell membrane, but we're not going in the right direction with the gradient. So we have to have ATP, or energy inputted, to help move it across the cell membrane. So again, the first one is transport of nutrients against the gradient or in the same direction, but at a faster rate. We can have presence of specific membrane proteins and the expenditure of energy. Okay, so any of these things will help expedite it. You don't have to have input of energy if you can move with the gradient, which is this passive diffusion. But if you're trying to move it faster or in the wrong direction, basically, you're going to need help with proteins in the cell membrane and ATP. Some substances won't cross the cell membrane, but are instead taken in through endocytosis. Endocytosis, we saw a little bit with viruses here, is when the cell will invaginate or engulf a food particle or a virus or something that they're trying to get rid of. The cell will enclose the substance in its membrane and form a vacuole around it. Amoebas and white blood cells will do this to help ingest potentially pathogenic particles. So they'll bring them in and try to like destroy them. If you're a white blood cell, if you're an amoeba, you're just trying to engulf something to eat it. And this is known as phagocytosis. Pinocytosis is going to be the endocytosis of liquids. So if you're trying to drink basically as an amoeba, you're going to engulf a pocket of water and bring it in, and now you have more liquid, not solid particulates. All right, so that takes us through the first half of chapter 6. I will see you next time.